Okay, so now we're going to continue with the third or the last part for our discussion in carbohydrate, which is the video number 3, the comparison of the structure and function for starch, glycogen, and cellulose. So this three is actually the most not most complex lah, but it's considered to be the macromolecule or the complex molecule that found in carbohydrate. They are polysaccharide. So when we're talking about polysaccharide, they are actually a link, or it is actually a repeating unit for many monosaccharide that join together by the glycosidic bond. So that is the polysaccharide. So when we're talking about polysaccharide, you also need to take notes about their characteristic. They are the very large and complex molecule. Okay, very large and complex molecule. And they do not same as the disaccharide and monosaccharide. When you taste the polysaccharide, they have no sweetness. Okay, they have no sweetness. They're normally insoluble in water because they are too large. They do not actually soluble in water. They do not form any crystallization between them. And they're actually a polymer. It is a very long chain of polymer, and they can be branched, they can be unbranched. Okay, the polysaccharide that you're going to learn is just the most basic type of polysaccharide, which is the starch, the glycogen, and the cellulose. The type of the polysaccharide that have only one type of monosaccharide that forming them. Okay, one type of monosaccharide that forming them. So the 4 example that you learned today is considered to be the homopolysaccharide. So there is also other type of more complicated polysaccharide which is not in your syllabus, like the chitin, the pectin, the hemicellulose, arginine, datrin. This is all the name that you were going to go through when you enter your semester 2. Semester 2, you're going to go through this name. The pectin, the hemicellulose, the chitin, they are also polysaccharide. But they are the complicated polysaccharide. They have more than one type of monosaccharide that forming them. They can be a combination of alpha glucose plus beta glucose plus fructose, and then they join together forming one type of polysaccharide. So that is heteropolysaccharide. So in your syllabus, you only learn homopolysaccharide, which is the easiest one. Amylose, amylopectin, both of them is a type of starch. And then the glycogen and also the cellulose. So what is the monomer that form all the molecule that I mentioned just now? So amylose and amylopectin, which is 2 types of starch, they are actually built out primarily by only one type of monosaccharide, which is alpha glucose. Same as glycogen. Glycogen also alpha glucose only. But cellulose is different. Cellulose is actually built out by beta glucose. Okay, beta glucose. So each one of these monomer, they are joined together by alpha 1,4 glycosidic bond. So this is the basic lah. For cellulose is the beta 1,4 glycosidic bond. Okay, because the first molecule is alpha molecule, so the type of bonding will be the alpha bonding lah. If they are beta glucose, join with beta glucose, both is beta glucose, so their bonding will be beta type of bonding. So, but if you notice, amylopectin and glycogen is different. Instead of alpha 1,4 glycosidic bond only, they also forming the alpha 1,6 glycosidic bond. So what is or where does the alpha 1,6 glycosidic bond come from? So that one will be in our next discussion. Okay, where you can find the amylose, amylopectin, glycogen, and cellulose is in the different part of the different organism. Plant. Inside the chloroplast, we have amylose. Inside the cell, plant cell, we have the amylopectin. Glycogen found inside most of the muscle cell, the liver, any kind of active cell that found in animal contain glycogen because they need it to store the energy. Plant store energy in the form of starch, so amylose, amylopectin is enough. But for animals, they need more energy, so they need a larger size of the molecule to store the energy, which is glycogen. And cellulose is found in the cell wall of the plant cell. They are basically just the structural material. So we go through. Now let us go through the structures and also the basic how the structure relate to the function of the polysaccharide. So before this, make sure you have this table with you when you want to do the revisions regarding to polysaccharide. So now we look at the amylose and amylopectin. So if you look at the diagram here, you're going to notice that amylose is actually a linear change. It is just a very long chain. It is only one change that's circular, that coiling together and then forming a complex molecule. So this is amylose. Because they are, if you uncoil this particular change, you're going to just getting a straight line. So because they only repeating the unit of the alpha glucose, so that's why their bonding is only alpha 1,4 glycosidic bond. There is no branch that found on amylose. Okay, different to amylose, if you look at amylopectin, you're going to notice that amylopectin is not one change. When they come to certain length, they were going to branch themselves into 2. And certain length, they will branch themselves into 3. So there are always a branching occurs throughout the formations of the amylopectin. So this kind of branching is caused by the alpha 1,6 glycosidic bond. So if you look at the structure in detail, you're going to notice that not only forming the alpha 1,4 glycosidic bond, but in some case, the carbon number 6 can also forming another bonding or glycosidic bond with others alpha glucose in the carbon 1. So this kind of bonding is called alpha 1,6 glycosidic bond, and they are also the reason why the molecule is branched. And what is the importance of by having this branch? So first, they will be able to form a larger size to compact more glucose in single macromolecule. So when they contain more numbers of glucose, meaning that the storage of the glucose higher, then more efficient as the storage molecule. Because when you have a storage room, how efficient the storage room is look at how many numbers of the item that they can store. So same go for the amylopectin. Amylopectin compared to amylose, they are more compact and they can store a larger numbers of glucose. That's it. So that's why starch have two types. One is the long change amylose. Another one is a long change and branches amylopectin. Okay. Finish about the amylose, we go to the glycogen. So what is the difference between the glycogen and amylopectin? I mean, both of them is built up by glucose. Both of them have alpha 1,4 and alpha 1,6 glycosidic bond. But what is the difference between the glycogen and the amylopectin? Yeah. So I think I scroll down, see whether there is a diagram to show. Okay, if you look at the diagram here, so amylopectin, if you look at the amylopectin here, they have the branch, but the branches is not compact. The branches is not that particular much. But if you look at the glycogen branches is happen very fast, and they are highly branched from one another. Okay, they are highly branched from one another. If you look at the amylopectin, normally in a very long end, only they have one branches occur. So compare a starch and a glycogen, you're going to notice that glycogens are more branch. So what is the advantage when the glycogen have more branch? Again, when they have more branch, meaning that more glucose can be stored. More glucose can be stored. Okay, and then with a higher branch, meaning that the molecule, the long chain of the molecule will become shorter lah. So they are not forming a very long chain. The chain go into the branch very fast. So when they go into the branch very fast, then the process or the surface area for the enzyme to actually break down the branch will be faster. So they are more readily for the enzyme to break down the molecule so that they can release the glucose faster to enter the cellular respiration and producing the ATP, the energy in the form of ATP. So when they're able to release the glucose more faster, provide the energy more faster, then they are more suitable compared to amylopectin to the animal. Because animal need energy to move. A lot of the process need energy compared to plant, which is only sit on one place and able to generate energy with two different modes. Photosynthesis also can generate energy. So that's why plant receive less energy. They need less energy. Animal needs a lot of energy. So they need to have a more complex molecule that can break down even faster. And when they do not need the energy, they can actually have the molecule which can store more glucose. And the glycogen is the one that fit this particular requirement. So last is the cellulose. Cellulose is a very long chain of molecule. They are linear molecule, do not coil. Compared to the amylose just now, they are coil. Cellulose do not coil. They are very long chain molecule, and they are actually held together by the hydrogen bond. So what is the advantage when they are held together by hydrogen bond is they will be able to form the microfibril, just like the thread. Okay, just like the thread, they were able to form the microfibril, and the microfibril will interwound between each other to form the cell wall. So this is how the cell wall is actually look like. It's actually a very long chain that can intervene to each other and make it to become a very strong supportive material. So that's it for the polysaccharide. And this is the basic on what is the thing that you have to know regarding to the polysaccharide. So we have done the video number 3. So hopefully you all can actually go through all 3 videos and prepare for your 3rd hour.